Hey team, let's continue our conversation on typical cellular activity. Okay, next is nutritional factors. This is pretty straightforward. If you don't have essential amino acids, you have abnormal levels of things, either too high or too low, then guess what? Your cell biology doesn't occur, your chemistry doesn't work properly, and you end up with conditions. This one's pretty straightforward, and honestly something you're probably going to end up talking about more in ortho, but it's something that's worth considering as a factor for cellular injury, because you're going to have plenty of patients who decide to play chemist and mess with different levels of things. Or honestly, this is getting more common, I would say, with all of our newer social media avenues, that people see things online and ask you questions. And it's good to be able to say things like, well, I think taking that much vitamin C might lead to issues with your GI tract because it's going to cause this to your cells, that sort of conversation. So what you're basically doing is you're applying your patho to keep your patient safe. That's really what we're thinking about when we're thinking about nutritional factors. Physical factors, exactly what you would think. So excessives in your physical environment, whether it's trauma, too much pressure, physical agents, too high or too low of temperature, or radiation. Specifically, we're thinking ionizing radiation or electricity, so like a shock injury. One thing to think about with temperature is that hyperthermia and hypothermia can both cause cell death. Hyperthermia or fire, burns, that's pretty self-explanatory, right? But with hypothermia, what's happening is that ice crystals and cell tissue are rupturing your cell membrane. That's actually pretty serious. So it's the old, you put a Coke can in the fridge and it explodes. Yeah, that's what's happening. So if you get too cold, ice crystals can form in a cell and lyse it. With hyperthermia, you're rupturing the cell membrane, right? Through the dissolution of the proteins, making up the different structures and breaking apart all their chemistry. Okay, with ionizing radiation, what we're really thinking here is radiation therapy for cancer treatment. So ionizing radiation is pretty strong stuff. So this is, in fact, it's capable of causing DNA damage and later genetic mutation. How it does this is initially through the process of stripping electrons from atoms that it's aimed at. So these types of radiation we're thinking of are X-rays and gamma radiation. So these can cause membrane damage and cell death. That's pretty serious stuff. You'll talk about that more when you get to your imaging course. But these are all things to consider, as well as just good old-fashioned UV radiation, right? Chemical factors. We'll talk about this more so as we kind of come to it with each individual tissue and organ system. But one thing to think about here is that there's kind of two ways this tends to happen. One is direct injury. So for example, if you were to hold liquid mercury in your hand, that it would bind to various amino acid groups and cause dysfunction of all these different functions of the cells that it's coming into contact with, right? And then there's stuff that's a little more subtle, and this is where we get into like our food safety and where we get our food from and how it's processed, as well as the different chemicals we have in our house, and then how our food is kept safe in the environment, whether it's like farm to table type logic, or how our homes are cleaned and things like that. So here's an example of something that's now banned, carbon tetrachloride. This is an industrial cleaning agent from the 70s. It used to be in old fire extinguishers. When it's ingested, the liver metabolizes it, and when it metabolizes the carbon tetrachloride, the thing that it metabolizes into causes damage. So this is a pretty common thing. Initially, the substance unto itself isn't necessarily a problem, but the way the body handles it can cause problems. So this is something why you'll sometimes see things recalled down the line, like, oh, we're now finding, so for example, that stuff like Teflon coated stuff, that's where they're talking about like forever chemicals and things like that. We now have evidence that actually maybe that wasn't such a great idea. So we can talk more about this as we go along, but you're going to see examples of this for the rest of your life, of something that they thought was safe and they later find out, oh, wait, the way the body handles that, that's causing cancer. It's always something like that. Okay, cellular accumulations. Oh, this is kind of interesting. There's 4 mechanisms in which this happens. Let's look at it through examples, though. That's a lot easier to kind of get a handle on. Okay, so the first one is abnormal metabolism. So if you have a lot of cholesterol that's building up in the body due to a lot of triglyceride intake, then eventually it's going to alter parts of your metabolism, because cholesterol is an endocrine active substance, right? We kind of forget that. But it can create, eventually, accumulations, right? And so that can affect your organs and their ability to function. So that's an abnormal metabolism. You've got too much of something in the body that's not getting handled like it's supposed to, and then it creates a problem. So because cholesterol unto itself is actually essential to life, right? It's an issue when you have too much of it. And too little is basically not a problem almost ever. Glycogen, easy to understand, right? That's taken up by your liver, your skeletal muscle, but it can very quickly become abnormal, right? So again, another abnormal metabolism. Let's jump ahead here to calcification. So if you have too much calcium in your bloodstream, too much serum calcium, eventually it's going to start to deposit. And when that happens, you end up with this sort of thing. So this is calcium buildup on a tricuspid aortic valve. So now these cells can no longer act properly, right? Which means the tissue can no longer act properly. This is actually a surgical situation. So we call this dystrophic calcification. So it's a real issue in a lot of cardiac type conditions when we're thinking of calcium buildup. This is a good example of abnormal metabolism because calcium is regularly in your system already, right? Like it's not necessarily a problem until there's too much of it. So now, how is there too much calcium in the system? That's a larger question to ask, right? But this just describes the phenomenon of buildup. You can also have, again, a defect in protein folding. So a classic for that would be neurofibrillary tangles and Alzheimer's disease. So the idea here is that something that should be a waste product that's getting kicked out, that's not happening due to different issues with how the protein is handled and its folding mechanisms. And so it builds up and you get a cellular accumulation situation in the brain, in the cortex. Let's see, pigments. The easiest one to understand here is the ingestion of an indigestible material. The classic is tattoos. The ink that's used is placed at a location in which, in theory, the body doesn't carry it away too quickly. It does very slowly, and that's how they fade. Actually, interestingly, there's like better places on your body to get it if you don't want to encourage fading so much, and it has to do with your lymphatics. So for example, if you're doing it where you have like really big lymph nodes, it actually won't last as long. So a classic for that would be the shoulder, because your axilla has a lot of lymphatic activity. So it's going to fade faster. But I mean, who cares? It's your body's art, right? But that's a classic example. So like less interesting, but more, I guess, medically relevant ones would be things like lipofuscin, which is linked to Parkinson's disease. So that can accumulate and it's not digestible. And so it gets stuck and it creates problems, right? Tattoos don't generally create problems. So yeah, we call it pigments, just as a heads up there, but really it's metabolic waste products. So for example, bilirubin, so if you've ever seen a baby that's born jaundiced or seen someone with liver dysfunction or something like that, and they look a little bit yellow, that's a buildup of the pigment bilirubin, but really it's a larger systemic issue, but you have an accumulation that allows you to make determinations about this person's health and what you're going to do with them. Okay, next is inflammation process. Okay, so there's normal inflammation. Any form of cellular injury is going to trigger an inflammation response. Inflammation's purpose is to restore structural and functional integrity as well as eliminate the problem, right? We have to remove whatever was, going wrong, get rid of all the waste debris, and then fix what was broken. This is like the entire content of your next lecture. We're going to talk about that in detail. In practice, what this really means is it's your initial defense against any sort of foreign substance, typically a pathogen. But I mean, it can be something like, let's say you get a splinter from handling wood. That splinter that's in your body, that's a foreign substance, right? So it can be something like that. Or it can be something that's a real bummer, like if you have a joint replacement and the metal that they put in your body, that's a foreign substance, right? So things to consider when inflammation is triggered. A response to cell injury, any type of injury, and it's closely linked with the healing process, and it can very easily go out of control, and so you can have chronic and detrimental issues. Again, next lecture. Aging. Okay, we're thinking normal aging here. So, in general, almost all of the cells in your body age and die and are replaced, right? Not all of them, and the ones that don't, very important, right? Heart, brain, skeletal muscle, stuff like that, right? There's stuff that does not replace, but it's a normal process for most of the cells in your body to, over time, degrade in their ability to function and to be considered something that the body needs to get rid of, right? Because it's no longer doing its job. Age in general as a biologic concept represents accumulated effects. So it can be genetic and environmental factors over time. And that's honestly how you can get disease, because basically we get worse. God, I wish we got better, but we get worse at repairing ourselves over time. So you're going to have an activity around the different forces that they think create aging, all the different things that have an effect, because this is a serious area for PTs. It's not wild at all to talk about this as a concept because we are steerers of exercise in the health field. So when we think about the role of exercise and its effect on cellular aging, it behooves us to understand what aging is. So we'll do this activity during class. Okay, let's kick over to another concept. This is reversible cellular injury. So we've talked about the ways that cells can get injured. Now let's talk about the ways that they change in the context of injury. We're going to have a lot of terms here, terms that we're going to use for the rest of the semester and honestly in the program, and you kind of want to be familiar with them. Okay, these terms are hypertrophy, hyperplasia, metaplasia, dysplasia, and then they're not cellular adaptations, but by convention we always teach these in the same bucket, atrophy and hypoplasia. Okay, let's start with atrophy. So this is a reduction in cell and organ size. So you can have a version of this that's not necessarily a concern. Let's say that you spent a lot of time in the gym and you built up really great biceps and then you got really busy with grad school and now the muscle's smaller. That's atrophy, but it's not necessarily a problem. We're talking about atrophy in a health context. We're usually talking about it where it is a problem. So this can be something where you have bone loss, muscle wasting such that it's a deep concern, such as in those images there. Brain cell loss, which means the brain, again, that's an organ, we kind of forget, but it's an organ, becomes smaller. You can have organ atrophy as well. These are all forms of atrophy in which, again, you have reduction in cell and organ size. That's a bad thing, okay? A lot of etiologies on how this happens, and we'll go through it step by step as we come into the different organ systems. So this is right here, this is osteoporosis. This is cachexia, you can see on one limb. And this here is the result of stroke over time. So this is atherosclerotic cerebrovascular disease. So basically this is vascular dementia, if you will. We'll talk more about that as we come, but in effect, this brain has not received enough blood in small ways, and over time it shrinks. So this is a healthy brain, this is an unhealthy brain from that angle. And you can see the difference in organ size. And then we'll talk about it in the context of cellular injury with accumulations, such as Alzheimer's disease. And you can see the difference in sizes between the typical or normal brain and the brain with injury. Okay, so there's hypertrophy. We want to distinguish this because it's important to do, right? You can have an increase in the size of the cell and the organ that's the result of increased functional demands. That's exercise-induced hypertrophy. That's a happy thing, right? That's not what we're talking about in this class. We're talking about pathologic hypertrophy. The classic example is going to be the heart. So what can happen over time is that a person has really high blood pressure. That blood pressure pushes back on the heart. In other words, the heart has to push blood out of itself into the periphery, out into the vessels through the aorta. And it's not able to do that because guess what? You have high blood pressure, it's pushing back. So the heart ends up in like a pushing battle. And in order to win, because it has to win, it gets bigger. So the left ventricle experiences pathologic hypertrophy. So we have here in the image is frankly a really terrifying amount of left ventricular hypertrophy in the context of very poorly controlled hypertension. This individual was 65 at the age of their passing. That is way too young for this to happen. I want to make sure that you have a good understanding of the difference between an athletic type hypertrophy and a pathologic type hypertrophy. So this is something you'll do during class in which you distinguish them. I'll go ahead and show you an image here. Okay, so we're using the heart because that's a really easy example. And let's look at an image here. So this is an echocardiogram comparing an athlete with a non-athlete and giving you a sense of difference. So remember that heart we saw earlier in which the chamber here was tiny. Here, let's look at it. Look at this. The chamber here is tiny. There's no room, right? Contrast this with an athlete's heart, right? This heart is bigger, but the chamber's intact, right? Like there's still room for blood to actually come in. So that means that this person's going to have higher stroke volume, right? So that's actually a good thing, okay? And then just out of curiosity, you can actually see the difference between different types of athlete and a non-athlete. We can look at this a little bit more during class, but what's really cool is that you have this sense that the chamber stays intact. Like the structural changes which occur in athletic hypertrophy are completely different than the structural changes we have in pathologic hypertrophy. Furthermore, they can go backwards, right? In other words, a Tour de France cyclist can stop being competitive and is going to revert back to the non-athlete anatomy. Whereas pathologically, that's less likely to happen, if not going to happen. More terms. Hyperplasia. This is an increase in the number of cells, leading to an increase in organ size. So let's contrast that with earlier. We had hypertrophy, which is an increase in the size of the cell itself, which then increases the organ, right? this one in hyperplasia, we have more total number of cells. That's the difference here, okay? So both organs get bigger. This one's because there's more cells. So this only occurs in tissues with cells capable of dividing. In the heart, that's not possible. Same thing with skeletal muscle. But in different other organ systems, that is possible. So a classic example could be somebody with prolonged estrogen exposure and the condition polycystic ovarian syndrome, and they have a thickening of the endometrium of the uterus. So that lining has more total cells, and that's why it's so thick. A non-pathologic example would be building a callus on your hand, right, from weightlifting and using the bar all the time. That's hyperplasia. You have more total skin cells at that spot, and that's why it's all rough and dry. So pure hyperplasia technically occurs in the result of hormonal stimulation or chronic kind of rubbing, like with gloves or something like that. But it's not always a happy thing, so just something to keep in mind, because sometimes this can occur. So for example, the classic here would be injury, right? And so an area that's injured over and over and over, you're actually triggering kind of like early cellular aging in that area. And as you age, you become more and more susceptible to cancer type changes. And so that's why I have here that hyperplasia can be a precursor to cancer. That's like years type precursor, right? That shouldn't be quick. That should be way down the line, but that's something to keep in mind. Metaplasia. Okay, this is the conversion of a cell type to another. So the cell changes in its morphology and its function. This is still reversible, surprisingly. But the example here would be, it's almost always kind of like a really bad situation. But the classic is a chronic smoker. Smoking causes metaplasia. You have a change in the type of cell expression inside the lung as a result of chronic injury from the smoke, right? And as well as the other like substances that are in cigarette smoke. So you have what's called squamous cell metaplasia of your respiratory columnar epithelium. So basically you get a cell swap out in response to chronic injury. However, if the smoker stops smoking, they're successful and they make that amazing, magical, awesome change in their life, and they are able to cease, like cessation is usually we call it, then actually the cell will change back. So we'll talk about that more when we get to POEM. This is actually a really cool thing you can tell your patients who are trying to engage in cessation, because we're usually supporters in that context. But yes, this is still reversible, right? So it's a swap of one cell type for another, but it's usually still able to change back, and the entire tissue does not swap out. So, but this is still scary stuff, right? It's definitely still scary. We don't want that. Dysplasia. Okay, so we have two things happening. Increase in cell numbers and altered cell morphology. That's going to cause a loss of the tissue's function. So that's the formal phrasing there, loss of histologic organization. This is a scarier one. This is pre-neoplastic. So that phrase neoplasm, think pre-cancer, okay? Pre-neoplastic, pre-cancer. This occurs in something that is chronically injured and keeps having to undergo hyperplasia or metaplasia. In other words, this is the smoker who didn't stop soon enough. So you can think of this as like the less scary version, both happening at the same time. So if you're getting hyperplasia and metaplasia, you eventually end up with dysplasia here in the middle, okay? And over time, you can end up with enough genetic changes occurring that you have something really scary. So this top image here, this one right here, this is an example of a pap smear. So for those of you who need to get a pap smear on a regular basis, this is what they're looking for. They're looking for dysplastic changes, abnormal cervical cells, because those cells there in the cervix are capable of dysplasia. They can increase in cell number and they can change in morphology, and that's what they can see when they put it under the microscope. So if they find that, that means you absolutely need to go back to your OB-GYN, right? Okay, so this is something to keep in mind. Your patients are likely to hear this sort of language. It's almost like we're kind of dancing around how scary it is, right? So the presence of some dysplastic changes, what they're going to say is, oh, we're just going to go in and take those out so they don't turn into something else. What that really means is that they're trying to get rid of something that can still reverse, right? We don't want to play the lottery here, because that's what we're doing. We're playing the lottery with, okay, is there going to be enough that all of a sudden this changes into something else? And something else it would change into is a neoplasm. Neoplasia is abnormal, unregulated growth of cells. This is the same thing as a tumor. Now, it can be benign or malignant, but at this point, if the smoker stops, the cancer doesn't go away. It's happening. So a neoplastic cell has lost most of or part of, or all of, sorry, so part or all of its original function. The appearance changes and it acquires a new function. So the new function here in cancer is pretty much often the same thing. It's invade other tissues, metastasis. So this is abnormal growth. It's unregulated. In other words, nothing's in control of it. The body can't tell it to stop. And if you have whatever the bad thing was that was causing the changes and it goes away, it's not reversible. So this is an irreversible change. Okay, there's two others we want to go over because we tend to talk about them in the same conversation. One is hypoplasia. This is the lack of a structure since birth. It's usually lack of a full structure. Aplasia would be the complete lack of a structure. It can be congenital or acquired. It's almost always congenital. And the classic here would be renal hypoplasia. So if you look here on this individual, this kidney is intact, but they're missing the other one. There's kind of like a little remnant here. So that gives you a sense of renal hypoplasia. The other one's not formed. So this is a person born with basically one kidney. And then we'll kind of continue the conversation on irreversible cell injury next.